morning, everybody. Thank you for being here. So, this might be a familiar image to you. These are the thorny-headed worms, the acanthocephalans, which have this hallmark feature, which is a proboscis bearing a variety of variable number of hooks. And these are used to attach to the specific habitat of the adult stage of the worm, which is the vertebrate intestine. It could be fish, birds, other reptiles, mammals. This talk focuses on one particular genus, which is Neochinorhynchus. Neochinorhynchus is a hyperdiverse genus with over 120 species worldwide. These are featured with 3 rows of 6 hooks. And these are typically in fish, but there are several species that have moved over to turtles as hosts. So as I said, they're highly diverse. As far as North America and the catastamids, we've heard about catastamids already this morning, they're really prolific in suckers. 15 of the 28 U.S. fish species are in catastamids. And so that's my focus, things like white sucker and chub sucker. In terms of life cycles, the general pattern is that there is a first intermediate host, which is going to be an ostracod, though this is not known for most Neochinorhynchus species, but the ostracod is likely the first host. And then so it's a 2-host life cycle, with the fish being the host in which reproduction occurs, so we call that the definitive host. Okay, so acanthocephalans, like many parasites, use the food web to complete their life cycle. This is an important aspect of their potential use as biological indicators. Okay. There are variations on the life cycle when you have fish that are larger, that are less likely to feed on ostracods. There may be extra hosts. You could consider these transfer hosts, paratenic hosts, like a smaller fish might eat the ostracod. In this case, a largemouth bass might eat that, and then sometimes largemouth bass eat one another, and so the worm can jump host cyclically up to another host. So these have some fluidity in their life cycle. But I want to tell you a little bit about the background, the impetus for this study. So Neochinorhynchus has been well known, documented in classic North American freshwater fish parasitology. That's a description from around 1919, very basic drawing, but kind of par for the day. And that same individual, Van Cleve, was part of this classic work on the parasites of Reyda Lake fishes. Other background, more modern work, includes morphological keys by amine, and then some work out of Mexico looking at the DNA sequence data. And so this is kind of the backdrop for this study. But as I did a fish parasite survey for the last 12 years with students at Otsego Lake, we ran into issues trying to identify these worms. They would not key out. And there were other issues with the genus, and as I said, a lot of this work is classic, older studies, most species described over 50 years ago, so no SEM data, scanning electron microscope data. And if you go to the literature, some of these worms occur in 8 kinds of fish all across the USA. And so it's kind of questionable large geographic variation, large host variation, relaxed host specificity, which kind of called my attention. I previously worked on stingray tapeworms and dealt with species boundaries there, so I kind of moved over to this. And the real thing was that there was no sequence data for any of the North American species. So this was a no-brainer as a choice for a master's thesis for Maggie Doolin, who was a previous grad student in my lab. I should mention this time, my co-author, Brian, is going to graduate this year. So if you know anyone who likes fish and worms, I'm looking for a master's student pretty desperately. Okay. So, So the current project was motivated by the fact that there's a lot of work to do. Okay, there's a lot of species to deal with, to ask questions of species boundaries. And this is a good model system for the phylum Acanthocephala, which only includes about 1,200 species. In other words, 1 in every 10 kinds of acanthocephalans is in this genus. So you can use knowledge about this genus to extrapolate to our knowledge for the order. So where do we go here? Our strategy was to sample new areas, but also sample old ones. So I refer to this as the type locality approach, where you go back to the original study sites where the species were described from, collect that kind of fish, the host fish, the type host, and then use those samples as a basis for morphological and molecular comparison. And so over the last 3.5 years, we've gotten a fair number of worms, and here you have a representation of their proboscides with scanning electron micrographs. And one of those had to be new. The lucky recipient of the raffle last night got a print of Neochinorhynchus bullockii, which is from white sucker from Reelfoot Lake, and that's the worm right there. And so I'm a systematist and a taxonomist. My interest is to go get the worms so I can study them in the lab, and I can do these kinds of phylogenies. And so we have a preliminary phylogeny here in which we can see a distribution of fish across various clades, and nested within this phylogeny is the turtle clade. And so already from this tree you can make the inference that the turtle worms were derived from fish life cycles, and they moved over to turtles at some point as their definitive host. And you can use these trees to look at morphological characters and look at the distribution of homology. But that's not my focus here. I want to tell you folks today about a big scale field effort and some unintended, surprising results. Okay, so we have done a pretty extensive effort over the last 3 years to cover a variety of sites. Some of them were not type localities, and so what this map shows, the dots are places where we tried to collect worms that were not type localities. If they're bigger, that means we were there more than once. So looking at the distance, you can see this was an effort. Who goes to Nebraska twice? Well, I'm from Nebraska, so that was me. The stars are the type localities. So there's 8 of them here. Okay, so 8 different type localities in 5 different states. And this map doesn't include, I told you it's a cosmopolitan genus, so I've also done work in Slovakia and in Peru to get these worms. But my focus here is North America and what might be going on in North American fishes. Okay, so I'll just run through some of these sites. First stop was in New Hampshire, Pawtuckaway Pond in southeast New Hampshire, and our target, the type host creek chub sucker. This is a catostomid that you don't see a lot of, and we were pretty happy to get 6, and 4 of those were infected. It was a lot easier to get the 30 white sucker in those streams, but those were infected with a particular species called Neochinorhynchus prolixoides, described in 1963 by Bullock. And so we collected those for both morphological and molecular study. That's the general plan in each of these trips. Then we went on to Oklahoma. This is Lake Texoma, and this is actually the same beach where Van Cleve and Timmons got the worm in 1952. We went back to the same spot. We gill netted for river carp sucker and found about a third of the 14 river carp sucker to be infected with Neochinorhynchus prolixus. So we were happy there and thought, well, we're going to just keep going like this, okay? But then we got to Biloxi down in Mississippi, and went there in 2017 and collected a nice diversity of fish, had nice hospitality from local biologists and friends, but did not get the black-tailed red horse until we returned there in 2019. We spent about a week doing a variety of collecting techniques. Still didn't get the red horse. I called the state, I plead. They said, we'll send you an e-boat. We got on the water. We had just broken a backpack shocker the week before. And so when the e-boat broke after 20 minutes, we really began to wonder if maybe we should change projects. But we did get 2 fish before the e-boat broke, both of which were infected, okay? So we did succeed in getting Neochinorhynchus buckneri from the type locality in southern Mississippi. So that was a nice result. And then we drove kind of a fool's errand. We got a lead from a state fish person that some ditch somewhere in Mississippi was a good place to get sharkfin chub suckers. So we went there, plunked down and right in there with a small seine. We got 4 individuals. We dissected one, nothing. Dissected two, still nothing. Third one, nothing. Last one, about a 20 centimeter long intestine. There was one worm in the last centimeter of the intestine. So we got there, okay? And that was Neochinorhynchus carinatus, right, shown right there. So that's southern Mississippi. But then I'm going to talk about failures now, obviously. And so in northern Mississippi, I'd been there with Doolin in 17 and then with Mullin last year. And this was a really interesting area to do work in the Delta. We worked with a fisherman who'd been there his whole life, had a lot of great stories to tell about the changing water bodies there, changes in the rivers. So we focused on the Yazoo, which is near the type locality. And in 2017, we looked at 3 different species of buffalo, 28 individuals total, which should have been enough to find a worm, but we didn't find any, okay? And I want to make this clear that when I say we didn't find any, we found other fish parasites, other acanthocephalans like Pomporhynchus, tapeworms, nematodes, trematodes, but not Neochinorhynchus. We went back there in 2019 to look for these 2 worms, one described in 31 by Van Cleve, the other in 49. And we looked at 18 buffalo, a couple different species. And this is important to mention here that we went in 2019 in August, which August in Mississippi State, that's an interesting time to go there. Bought a lot of ice. We had been there in 17 in May. And so in a lot of the species of Neochinorhynchus, there is some seasonality pattern to the life cycle. So the question of why didn't we find it in May? One answer would be, well, maybe it's a season when the prevalence is lower. So we thought we'd go at a pretty different time, fairly different time, August. But then again, we found no Neochinorhynchus. And so here we are doing work in Greenwood, great place. We didn't have a lab. It's too hot to work in the daytime, so you do kind of these suspicious looking things like open up the tail, you have your rental car, line it with a tarp, and go through a bunch of large buffalo, a couple hundred pounds of fish, get the intestines out, and then dissect the intestines in the hotel room all night. Meanwhile, you disposed of the fish. I won't get into that, but we did do that, in a way. So then we went up to Reelfoot Lake, and Reelfoot Lake has been a fisherman's mecca. So this is in Tennessee. Maybe you've heard of Reelfoot Lake as a bass or crappie fishing destination. Reelfoot Lake was formed in the mid-1800s when the Mississippi River flowed backwards because of a massive earthquake and backfilled a swamp, which are the cypress knee swamp, which is a beautiful fishing habitat. There's a song about the creation of Reelfoot Lake. And so we went there in 2017 and looked at fish provided by the state, and then we went again in August of 19. So this is a May sample, and then this is a August sample, okay? And we were looking at buffalo, different species of buffalo for Neochinorhynchus sturgiosus, which Van Cleve found in 1949. Okay, so I was there in 17 and 19, we didn't find any, okay? And so had there been changes between 2017 and 1949? Actually, I would tell you that there's probably been changes since 2003 because of a colleague who went there in 2003 and found a prevalence of 1 in every 3 buffalo. Unfortunately, that colleague did not save any for DNA work. And so that was 2003. We're there in 2017. The fish are there, the population seems good. They've got other worms, they're eating other invertebrates, getting those parasites, but no Neochinorhynchus. Okay, so the question is why? Is it really not there? I'm going to argue that it's not there. And it's, of course, harder to prove the absence of something than the presence of something. But given the numbers of fish we've looked at, given the known prevalence of other Neochinorhynchus, we would have expected to find it if it's there. Okay? When we went to Reelfoot Lake last summer, people were really happy to see us. They said the tourism has gone way down. Okay? And that has to do with this. And so Reelfoot Lake, as I said, was formed by the Mississippi River. When there's floods, which you could talk about global climate change and the higher frequency of floods now, enables silver carp to move over into Reelfoot Lake. And in the last few years, actually since 2017, the silver carp went there and has changed the fishery. So crappie fishing is done, and even getting the nice big bluegill is harder now. Really all you catch is catfish. So could the introduction of a fish like this, which by the way is a horrible host, no worms, 10-foot long intestines, like purgatory going through that thing. Could that have changed something? Well, the buffalo populations seem okay, but could the introduction of the silver carp somehow have changed the food-web interactions such that buffalo are no longer getting the ostracods? Okay, so that's my guess, but we don't really know. And so invasive species can shift ecosystems and therefore life cycles of parasites. And in conclusion, I would say that we have been successful in some type of localities. So these worms are, continuing to carry out their life cycles in a variety of locations throughout the country. But for the 3 type localities that we visited twice, we have some degree of confidence in saying that those species are extirpated from their type localities. That's the major conclusion here. Well, is that good or bad? Well, parasites do harm to their host. You saw those hooks. Those cause all kinds of problems, hemorrhaging in the intestine. But if you lose a parasite, that is some kind of a sign of a change in the ecosystem. So I'd argue it's a negative feature. And so we'll see what happens there as we continue to use this type locality approach. So the landscape is changing, the fisheries are changing, and we see that reflected in the worms. Okay? And so the acknowledgments, I can't talk about everybody that made this possible. There are a lot of people, some of them are in this room, who helped us acquire fish, fellow biologists that I knew through parasitology, but also state fisheries people that I got to know via email and were incredibly hospitable throughout the South and Virginia and all over. And I have to point out Maggie Doolin, who did talk at AFS 3 years ago, her master's degree thesis really got this project going, and now we're continuing it with various undergraduate projects. So thank you for being here, and I'll take any questions if there's time.